I did make, oh, okay, got it. Okay, so we got it there. Okay.
I'll do a quick introduction for you. This is Lisa Ridnour. She has been here on University of Iowa campus and is an alum, so she's familiar with what you all are studying and being here at University of Iowa. She's currently a staff scientist in the Cancer Innovation Laboratory at the Center for Cancer Research at the National Cancer Institute, and she will share a little bit with us about her career path. Um, and where she's been, where she's headed, and then questions are open thereafter. So if you've got a question for Lisa, please unmute your mic and ask, and ideally show your video so she can talk to you face to face. But if you're in a noisy area and you need to put it in the chat, I'm fine reading that aloud too. So welcome, Lisa. Thanks for being with us today.
Hi. So it's fun to talk to people from Iowa, as always. So I don't know if I need to swap my screen or not. Can you see what I'm sharing?
If you can swap, that would
All
be.
right, let me try to. Let's see. Did that work?
Nope.
Nope. Okay. Let's see. Okay, let me cancel out of this. Let me stop sharing and okay. Okay, so let me do this. Okay. And how about that?
There we go. Yep.
Okay. All right. So I'm going to back up here. So I started at Iowa.
Lisa, I think you froze.
Become an adult, and so I decided to go to graduate school. And so, I got into his program. As you can see from my screen here, I'm still an avid Hawkeye fan, so I tune into a lot of Hawkeye sports. And this year, I got really interested in the women's basketball because they were so fabulous, and that was a lot of fun to see them, go all the way to the national championship game, and that was great fun. But anyhow, when I left Iowa City, I went to St. Louis where I worked for Doug Spitz at Washington University, and I got my postdoc from him. So we were looking at nitric oxide associated stress responses in his lab. So I was looking at the role of glutathione and peroxidases in the development of stress responses to nitric oxide. When I left his lab, I came out here to the East Coast and I joined the National Cancer Institute, where I worked down in Bethesda. And I worked for two different groups. Now I'm in Dave Wink's lab, and I have been with Dave Wink since 2003. So I've been with him for 20 years. And we moved our lab up to Frederick back in 2012. And so this is a nice little picture of Frederick. And Frederick is really a beautiful, quaint East Coast city. And, so for people coming up here, sometimes they look around and wonder, what do you have to do in Frederick? But there's really a lot of interesting things to do up here. If you like the outdoors, here is a picture of me, rock climbing. I did that when I turned 50. I decided I needed to kind of like reinvent myself. So I decided to learn how to do rock climbing, and that was very a lot of fun. If you like good food, Frederick has a lot of wonderful restaurants, and downtown is very artsy. There's a huge community of artists, and people enjoy outdoor activities, and there's a lot of history out here, of course. So, there's lots of things to do, and it's really a very, very nice place to live. When I joined Dave's lab, one thing about Dave Wink is Dave is always interested in something new. And the latest thing that he became interested in is, spatial imaging. And so for the past few years, we have kind of expanded our laboratory to use spatial imaging to study roles of, nitric oxide in the disease progression of breast cancer, and how the immune response is affected and how it's modulated by high levels of inducible nitric oxide synthase and COX-2. And so you can see a little bit of the data here that we have shown, and it's really been a wonderful opportunity for us because working for Dave, we're always learning something new, but we have also moved this field forward significantly, I think, because what this work is showing is that when we have high tumor NOS2 and tumor COX-2, it's associated with a very, very poor survival in humans. And also mice that have aggressive 4T1 tumors, these are triple negative breast cancers. These mice don't survive very long, but when we target NOS2 and COX2 in these tumors, we can actually get nearly half of these mice to survive, a second challenge to the same tumor. And the interesting thing about that also is that our work it's contributed to a recent clinical trial where people that had drug-resistant breast tumors and they had a very, very short time to live, they were given a NOS inhibitor with taxane, and these I think 25%, close to 25%, there was an overall complete pathological response in 25% of these women, and there was an overall response of nearly 46%. So that is really, really good, for these tumors, because most of these women don't live. And so a great thing about being out here at the NIH is that we have a lot of resources. And so, like I said, Dave got interested in doing, all of this imaging work, and we had no idea how to do it. We didn't have that capability, but we developed collaboration with an imaging person up here in Frederick, and he helped us to get started, doing these imaging analyses. And so we have looked in mouse tumors, mouse liver tumors, mouse breast cancer tumors. We have looked at human breast tumors, human liver tumors, and it's expanding. We're getting melanoma tumors, and he's interested in pancreatic tumors. But there's a lot of work to do, and there's a lot of resources up here, and a lot of different expertise. And so it is really a great place to be if you want to do science. And the way I got here was really, it wasn't from just applying for a job. It was through networking and through people that I knew. In fact, the way I met Dave was, we were co-authors on a paper that I did when I was in St. Louis, in Doug's lab. We did a methods paper where we quantified the levels of nitric oxide in cell culture media. And so when I moved out here, Dave used to come down to the oncology labs all the time, just kind of wander around to talk to people. And so, he didn't know me because we had not met personally, but, we had collaborated. So I introduced myself and, we just would start talking in the hallways and whatnot. And then, basically he offered me a position in his lab because, I started out working in a different investigator's lab. And so I've basically been with him, for the past 20 years. And it's turned out to be a really, really wonderful opportunity for me because we've done a lot of great work. We help each other out quite a bit. One thing about Dave is dyslexic and he has attention deficit disorder, so he doesn't write well, and so that is a big part of my job. So I help to write our papers and our site visits and things like that, and we've worked well together for many many years. But you know I think for most of the jobs that I have taken, I haven't really, like I said, applied, or looked, in the newspaper or online. It's just been networking and, people that I have, come to know, through my contacts, through Spitz, um, through Larry Oberly, so I think that that's been a fun part of my career as well. But anyhow, if anybody has questions, any specific questions about coming out here and what it's like to work out here, please go ahead and ask them. Everybody's pretty quiet.
I can ask a question to get us started, Lisa.
Okay.
What would you say as a staff scientist at NIH? What's your typical work day or work week like? Or if that was a job somebody was looking into, what might they expect?
So, I do a lot of different things. I run an animal program here. So, I will prepare cells and things like that for injecting them into mice. We also have a radiation facility, which I run up here. And so when we're irradiating mice, I usually go and do the irradiations, but I also train people, to operate our irradiation cabinet. And so hopefully, they'll be taking that aspect of that part of our work over. But, like a typical day for me, I manage our lab. So, I help train people to learn how to do experiments. I keep the lab going just by buying, I purchase all the materials and reagents and supplies and things like that we need. I'm involved in, meetings with students. I help them with their work, when they have, problems, with experiments, or, I help to teach them, how to do specific techniques and things like that we're interested in. Sometimes I get to work from home. So because I do a lot of desk work, associated with the writing aspect that is a big part of my job, I can work from home, a fair amount. Usually I do come in, but if I have to work late, I do. If I have to work on weekends, I do. But typically, I am not, I don't work weekends and I don't work late at night. Usually, I usually am gone by probably 6 or 7. And that's with the day starting a little bit later. Our day starts, like my day starts, between, 9 or 10. So it's, I think I like it. It's not a typical 9 to 5 job. But I like it because I can set my schedule, and I think it's good. It works well for us. So um,
Very helpful. Trainees, what questions do you have for Lisa? everybody shy today?
I guess. Yeah.
Hi,
Oh,
hi, Lisa.
Serena's got one.
Okay.
I have a question. I'm considering um taking a fellowship at the NCI. I kind of just wanted to know how if you had to choose a different career path, maybe within the NCI or maybe within industry, would you, or are you um content with your career path as a staff scientist?
You know, I like I like my job. I like the job that I have. Again, for me, I think it's very satisfying. I mean, this work that I'm showing you, to me, this is very, very satisfying. The fact that our work has it's a big part of this clinical trial that was just completed. This was a small clinical trial, but the results were very positive. There was good survival associated with the treatment, and this, to me, I find this fascinating, that we can target two enzymes that are just in the tumor. We can target two enzymes and we can get this kind of improved survival, just by targeting these two endogenous enzymes, right? And you can target them with drugs that are clinically available. So I find this very satisfying, that I firmly believe that the work that we're doing is going to help people down the road. And so no, I wouldn't change my career path. However, if bench science is not for you, because it's not for everyone, there are still a lot of opportunities out here, because the NIH is a huge place. People go through postdocs and they end up getting positions in, like, the office of the director, or there's just a lot of different opportunity that's available out here with the NIH. So I think it's a really good place. And if you're interested in doing a post-bac, there are different websites that you can apply online for, and then you would submit, a resume. And if there are specific PIs that you're interested in working for, you can submit it directly to the PI, because we get resumes from students, a fair amount of time. In fact, one of your students, Elise Femino, do you remember Elise Femino? She worked for Mitch Coleman, and she is out in our lab right now. She's doing a post-bac. So she's been here for a year, and she actually is doing a lot of this imaging analysis work. So she's, staining tissues, she's doing the imaging, and then, she is involved in imaging them with the software that's available to us. So she actually gave a couple of talks already. She went to Maine with us and got to participate in a meeting in Maine, and she got to go out to Ventura, California, where she was selected to give a talk at a Gordon conference. And we asked her to give a talk in our CIL Monday seminar series, and she gave a very nice talk for there. So if you're interested in science, I think the NIH, and the National Cancer Institute is a great place, to come out to and get your feet wet. And there are plenty of, websites that you can look at and apply on. If you have some specific questions, you can certainly send me an email, and I can, help you, at least as much as I can, by, providing, information and contacts, people for you to contact. So
Thank you.
you're welcome. I did actually go to a startup company before I came out here to the NIH, and I worked at the startup for, well, I guess it was a little over a year. And these people, I thought that they were really a great fit for me because they had developed a superoxide dismutase mimetics, so that was coming out of Larry Oberle's lab and being a student of Larry Oberle. That again was another networking contact that I made. And, I had a job opportunity as a result of it. But unfortunately, because they were a startup company, they failed. Not all startup companies survive. In their case, they were getting money to support their work and everything, but this person that they hired as the president of their company came in, and I think from the sounds of it, from what I understand, he kind of took the money that they generated and funneled it into another business. But anyway, that was what I thought was going to be a good opportunity for me at the time, but it didn't end up working. And so again, through other contacts, I knew David Gaius from my days at St. Louis at WashU. David was a PI there, and he left WashU and he came out to NCI. And he knew of a lab that was going to be starting up because the PI was moving there as well. And so, I sent my CV to that person, and I, got a job, in his lab until I decided to make a change and jump over to Dave's lab. I think Dave's lab has been a great fit for me. And, he's really a great person to work for. So,
what other questions do you have for Lisa? Travis has one in the chat. What do you feel prepared you most for the job that you're in now? Training during your PhD postdoc, experience at the startup, or other experiences?
People have described me as a jack of all trades. And so that sounds kind of funny, but there's probably some truth to it. And this might sound funny, but, when I was younger, I would be a little bit nervous, and maybe, afraid of the unknown, which, it may sound weird going into science, but if somebody asked me to do something, I didn't want to fail. And so I always just jumped in with both feet, and I worked as hard as I could, to produce data. And so I say that because, when I did take these different jobs, I actually had to learn some new techniques, things that I didn't know, to generate, data that was required to ask questions that, they wanted to answer. For example, when I worked for Phil Toffelon, he was interested in, he studied astrocytes. So I had to learn how to culture astrocytes from neonate rats, and, something I had never done before. So the protocol he gave me seemed to be a little bit funky. So I actually had another friend from WashU, and I contacted that person, and she kind of, helped me to troubleshoot the cell cultures, and I was able to develop really beautiful astrocyte cell cultures. And then another thing that Phil wanted me to do was he wanted me to set up mitochondrial enzyme assays. And so I learned 4 different mitochondrial enzyme assays, which, I hadn't run before. I had done, a fair amount of enzymology, of course, because I worked for Oberle and Spitz. And so we assayed for superoxide dismutase and catalase and, GPX and all of those things. So I, learned how to run these other enzyme assays. And, I developed some pretty nice data sets from that. When I jumped over to Dave's lab, we were kind of interested in looking at MMPs, matrix metalloproteinases, and I developed this interest based on studies and work that a friend of mine at Washington University had conducted, where, he looked at how post-translational modifications affected the enzyme activities of MMP. I think he was looking at matrolysine, MMP7, and MMP9 and MMP12. And so, I had, through my reading, I had read that NO could modulate some of these MMPs. And so that's one thing that Dave and I would talk about in the hallway a lot, so I started studying MMPs, one of the first things, and extracellular matrix proteins when I started working for Dave. And then, after that, a few years later, again, we got interested in the imaging studies. And so I learned how to do, these images and image analyses. And I think it's just, being willing to try new things. And so I think that actually has been my strength, in moving through my career. So I'm kind of at the end of my career. I'm in my 60s right now. And so I'll probably be retiring in a few years. But, I think that if you guys are interested in science, definitely, come out to the NIH, come to NCI, because there's a lot of opportunity. There's a lot of resources. You can do a lot of different things out here. So.
Good advice. You've shared about the rewarding parts of your job. Are there areas that have been challenging for you?
Yeah, I mean, all of it. learning, all these new areas and things that, I haven't done before. When we moved out here, now we're in an immunology program. Immunology, I find immunology to be even harder than matrix biology. It's like learning a new language again. I thought that of matrix biology. It's not linear, but really neither is redox biology, because that's one thing that we, identified in our studies with NO. We would find these biphasic responses, concentration responses or concentration dependent responses to NO. And it's a very similar thing, with matrix biology, with immunology. So yeah, I think that there's been a lot of hurdles that we've had to jump over. But, I think that we've done well. We've, as I said, I'm very, very happy, with the work that we've done. And I definitely think that it's going to help people down the road. In addition to the cancer work that we've done, we've been involved with a collaborator of ours who studies Alzheimer's disease. And while, we've shown that high NOS2 promotes disease progression of breast cancer and many other cancer types, our work with her actually demonstrated that high NOS2 seemed to be protective against the disease progression of Alzheimer's disease. And one mechanism that we identified was that when NOS2 was present and you had NOS2 derived NO, you had higher MMP9 activities, which could chew up the plaque in the brain. And so we got a nice little paper out of that. I think we published that in neurochemistry, or the Journal of Neurochemistry. And so, but again, maybe hopefully, the work that we have done can also, be helpful in for Alzheimer's patients. Another interesting thing was, so she had a mouse model, and we, tested this in her mouse model, and we found that her mice that had high NOS2, there was more MMP9 activity in the brains, in the hippocampus of these mice. And so we developed an assay using mass spec, where we developed an antibody that recognized a peptide that associated with MMP9 degradation of the plaque, and we characterized it by mass spec. And then we developed an assay where we IP'd out this peptide fragment in the mouse brain, but also in the brain of human healthy controls who had passed, as well as people who passed of Alzheimer's disease. And we found that the healthy people had 2.5 times more MMP9 activity than the Alzheimer's brain. And that was consistent with the mouse model that we were studying. So, it's really been great. I've been really fortunate because, I get to work with a lot of creative people, and, we just, I've learned the things that that I've been asked to learn, to answer questions that we wanted to address. So again, it's a great place. You can do anything you want out here. There's a lot of toys. So,
any other questions for Lisa? If not, I will wrap us up with one. You kind of touched on it, but would you share what your work-life balance looks like? Would you say that it's where you want it to be? Or was that something you had to earn a little more of as your career went along?
I think that, I have a pretty good work-life balance. I have had, I have friends. If I want to go out and do something, there's a lot of things that I can do out here. In addition to, fine dining, I like to dabble in cooking. And actually, this is something that I, cooked at home. That's a bouillabaisse, so lots of different fish and seafood, in that dish. And so that was really yummy. I I think I have a pretty good, work-life balance. And so, but again, some people are obsessed with their job and that's all they do. I am not one of those people. I'm not one of those people that, eats, sleeps, and drinks science. So, maybe I might be a little bit healthier, you know, because of that. So also, I think that's good because, I think maybe you can maintain your stress levels a little bit better. Make sure you get your sleep, make sure you get your exercise. And, I think those are some good, good habits to keep. So
it's all about balance.
Yeah.
Yep.
So.
All right. Well, we appreciate you taking a bit of time to chat with us, Lisa, and answer some questions. And is it okay if I share your email address to folks?
Yeah, sure.
If they'd
Absolutely.
like to reach out?
Yeah,
Okay.
sure. And, they can, send an email and I'd be happy to help them with contacts. And, maybe even if somebody wants to come out here, maybe we can, give them a job as a post-bac or something like that, you know, kind of like Elise. Elise is doing a great job, so I'm glad Mitch told me about her. So
very good. I will, if you've shared anything today, it was the importance of networking. So we'll make sure that your information is shared with them so that they can network with you after this as well.
All right, great.
All right. Thanks, everybody.
Bye.